So good evening, I am Francine Béard-Cohen. I am an ophthalmologist and I'm the founder of Eyevensys. Eyevensys is a clinical stage company that is developing a new innovative platform delivery system for any therapeutic protein to be delivered in the eye. So Eyevensys is a clinical stage non-viral gene therapy company that targets ocular disease. It's an innovative strategy that allows the sustained expression of any therapeutic proteins inside the eye using a minimally invasive procedure. The proof of concept of our technology has been demonstrated in an ongoing phase 1-2 trial in the United States as well as in Europe. And now we are accelerating 2 projects for 2 different type of patients and 2 new proteins. What is Eyevensys? Eyevensys is a technology that is made of 3 components. A plasmid DNA with a proprietary plasmid backbone and allows to incorporate any gene encoding any therapeutic proteins without limiting size. And we can also insert even 2, 3, or 4 proteins, genes at least. The second component is a device. So it is applied on the eye surface under topical anesthesia. And the third component is a generator. This intends to incorporate inside the ciliary muscle cells a plasmid encoding a protein. And once it is incorporating using electroporation, the ciliary muscle becomes a biofactory that secretes any therapeutic protein inside the vitreous, as well as in the tissue of the posterior segment of the eye. The proof of concept of this technology has been shown using different proteins in different animal models in preclinical studies. In humans, I606 delivered a soluble receptor for TNF in patients that were end-stage uveitic patients that were legally blind, actually. 16 patients had been enrolled, and there were 3 cohorts of patients with escalating doses. This intended to show the feasibility as well as the safety of the procedure. And it showed that it is feasible under topical anesthesia within a few minutes. But interestingly, in the 3rd cohort that had the highest dose of plasmid, we could see in 2 of the 3 patients interesting biological signals of efficacy. As you see, this patient with retinal vasculitis gained 12 letters, and this was associated with reduction in his macular edema. The patient here with a Burchard retinopathy also had a gain of vision associated with reduction of his macular thickness, but the study was lasting 6 months. And as you can see, after 2 additional months, there was still a reduction in macular edema. And this witnesses that there is still protein expressed in the eye up to 9 months after a single treatment. Indeed, this strategy is intending to deliver and to produce protein for about 6 to 9 months, but not continuously, so that there is an exit strategy and the patient is going to be retreated and followed up. Now we are developing 2 new treatments for 2 types of patients. The first one is I809. It's a dual gene plasmid containing the genes of 2 proteins. One is aflibercept and the other one is decorin. Is it intending to treat patients that have persistent fluid after optimal treatment with anti-VEGF? Indeed, not all the patients respond to anti-VEGF because VEGF is not the only factor involved in wet AMD. So this will be additive or alternative therapeutic approach to VEGF inhibition. Why? Because decorin have strong anti-angiogenic effects independent from VEGF, but also anti-inflammatory and anti-fibrotic effects. So we intend to increase the percentage of responders with wet AMD and provide anatomic benefit of a reduction of leakage induced by anti-VEGF. We showed that Eyevensys technology was superior to reduce leakage in animal model of CNV as compared to injection of aflibercept. The second strategy that is developed now by Eyevensys is intending to treat patients with retinitis pigmentosa, whatever is their gene defect or mutation. Indeed, it was shown that iron accumulates in the retina of patients with any retinal degeneration, and this iron is inducing a strong oxidative damage that kills photoreceptor cells. And we are producing now, with the Eyevensys technology, transferrin. Human transferrin is the natural iron chelator. Not only is it chelating iron, but it's having additional neuroprotectant effects on patients with retinitis pigmentosa. It has been shown to be effective in animal model and safe to slow photoreceptor degeneration and loss in various animal models, including animal model of retinal degeneration. But it could also be applied for patients with dry AMD as well as glaucoma. We showed in the rabbit that we are able to produce after a single treatment efficient levels of transferrin in the aqueous in the vitreous and the retina and choroid for a period of at least 6 months. In the pipeline, we have 606 ongoing on patients with escalating dose. We have 611 using transferrin for neuroprotection, first retinitis pigmentosa, but other indications, and I-809 with patients with wet AMD. The company is managed by Patricia Ziliox, which is the CEO and president, Ronald Begage, who is the CMO, Thierry Bordet, the CSO, Gina Pinto, who is the finance and administration, Alan Versky for device, and Helen Mortimer for clinical operation, as well as our board of directors and our investors. I thank you for your attention.